Hey everyone, today we are going to be talking about viral latency, a really important concept and something that's pretty unique to viruses. Now, there are some parasites, some higher order parasites that can kind of hide out inside of people in like a cyst and then reactivate. And that's very similar to what we're going to see with viral latency. But there are certain viruses and certain families of viruses that engage in this a lot. HIV being the most common one, also herpes viruses, arguably the most successful viral family on the planet. So let's get talking a little bit about it. We're going to start with some diagrams from Epigenetics and Genetics of Viral Latency, which I have the link below, and I'll post that below. The main characteristics of a latent virus are that it's going to establish a long-term persistent infection. Okay, we call those persisting viruses, at least the genomes of those persisting viruses, that's the reservoir of the virus. Okay, so those reservoirs are not actively making virus. Okay, that would be a low level of infection. Okay? When the virus is not actively making virus and it's just kind of hiding out there, not really doing much, though we're going to learn a little bit later that they may actually be doing some things. That's the viral reservoir. And so in HIV, we talked about this very often. So you would ask, what's the patient's HIV reservoir? Those are the viruses that are just hiding underneath. And so that term got thrown around all the time. Now, what can happen from those viruses in the reservoir, they're not actively replicating, they can reactivate to start replicating. And that's a really important idea too, to actually go from a latent state into a reactivated state and the factors that are engaged there. And we're going to talk a little bit about that. We do see this concept in other viruses in lower order organism viruses, so like for bacteria. So bacteriophages will engage in something that gets talked about a lot in basic biology classes and microbiology classes, the concept of a lytic cycle and a lysogenic cycle. So we're going to be talking about the mammalian version of that. So a lytic cycle is what we typically associate with viruses. It gets in there, it takes its viral genome, embeds it into the host genome, and then it grows new baby viruses, and eventually it explodes. Now in mammals, we actually see more often, the cell will eventually die, but they don't often just build up, and then explode all the viruses out. That's not typically how we're going to see that. Most of the viruses that we're going to deal with have membranes. They have envelopes. They're enveloped viruses, and so they're going to steal that envelope from the surface of the cell. Bacteriophages don't usually do that. They're not going to steal the thick, hard cell wall from a bacteria. It's just not something that they do. Now, the lysogenic cycle that you may have encountered before, that's more the latent version in a bacteriophage. So if mammalian viruses, some of them can undergo latency or enter a latent state, then some bacteriophages can enter a lysogenic cycle. And that's just where we see their viral genome gets integrated, a very important term, I'm going to use that a bunch. It gets integrated into the host cell's genome. It's going to hide there, and the key to that is its viral DNA embedded within the host DNA. And so the host machinery, as long as that virus genome gets in there, the host replication machinery, all those DNA polymerases, they have no idea that there's a virus there, so they just copy that viral DNA just like they do the host DNA. And so you end up with all the daughter cells also having that virus. And so you get this persistent viral infection. Eventually, due to specific factors that people are still studying today, that virus can then reactivate and enter the lytic cycle. And then you have just a normal lytic cycle, and new baby viruses get made, and then it expands out. All right, so let's keep on moving. We're going to talk a little bit, this comes from that same paper. These people, there are a lot of scientists that have studied latency, and this idea of the host pathogen conflict, it's a really important idea. The idea that humans, or we're going to say humans, but mammals in general, or all creatures that are living, have been fighting this evolutionary battle over however long you think this battle has waged. Millions of years, it doesn't matter. There's always a push-pull between the host and the pathogen. So viruses infect the host, and then the host develops some kind of resistance, and then the virus tries to resist that. Sometimes the virus wins and can reproduce. Sometimes the virus loses, and we get rid of it. Latency is arguably a way that there's a draw. Okay? Now, I don't necessarily completely agree with saying that all latent viruses that are able to establish latency, that is a draw. I think that's kind of an oversimplification, not to attack the people who made this because they're way smarter than me. But it's more just that's the life cycle that this virus has chosen. I would argue that establishing a latent infection is not a draw at all. It actually allows the virus to hide and better infect more people. Why not hide and wait? Let's kind of lay low so that the host immune response kind of chills out and the cells don't know what's going on. The cellular immune response doesn't know what's going on. And then reactivate and kind of sneak out there and take over. Sounds like a good idea to me. A lot of viruses do it. So, but that's where this idea comes from. But the point I'm trying to make here is that the purpose of latency is to wait until there are optimal conditions. Because always remember, every living thing, which viruses arguably are not living things, but they do behave, they reproduce like living things. The purpose is to make babies, right? I say that a lot. The purpose of living things is to make more babies. Viruses, in this context, are living things then. They want to make progeny. So, in this case, they're going to wait till the optimal conditions because that's going to mean that most children will be successful, right? That's what our goal is. I want the most children to be successful. And for a virus, that means they're going to go on and reinfect new host cells or a new host that has host cells. And so that's their goal. And so some viruses use latency to take advantage of that. Another diagram, great diagram from this article that's going to talk about the episomal versus proviral integration. And so I'm going to jump to my zoom out here and let's talk a little bit about, I dropped my marker, proviral versus episomal latency. There are two types of latency. Okay, there are episomal. It's episomal. Okay. And there is proviral. Okay. So I'm going to draw both of those. So there's my cell, and I'm going to make a really big nucleus. Okay. So that's my nucleus. I apologize, it's making kind of a banging noise. Okay, so there's my nucleus. I drew it really big because we're going to draw a couple things. I've got two colors here. I'm going to do episomal in blue. So in the episomal concept, the virus is going to enter the cell. It's going to get its genome into the cell, right? That's the goal of a virus. It needs to get its DNA or RNA into the cell. With latency, we're going to usually be dealing with DNA viruses. I can't actually think of any RNA viruses, at least any that will integrate their RNA. That just doesn't make sense. HIV is an RNA virus that converts its RNA to DNA and then integrates that. So yeah, it's tit for tat, but whatever. So with your episomal latency, what's going to happen? So this is like your herpes viruses, your cytomegalovirus, chickenpox, all that kind of stuff. We're going to have our host, or our viral DNA. It's going to come in, and we're going to enter the nucleus. Now we want to be inside the nucleus, okay? Because that's where all our DNA replication machinery is. That's just the right place you want to be usually if you're a DNA virus. There are exceptions, but we're going to kind of ignore exceptions for now. So there's my viral episome. We're going to call that an episome. This is different from the circular proviruses that sometimes we see in HIV. For those of you keeping score, don't worry about those. Those are something separate. I'm going to draw my host DNA. Okay. It's obviously a lot bigger than that, but there's my host DNA, and it's hard to tell, but I'm going to, with episomal, I'm going to leave it near the host DNA. It's just going to hang out there. It's going to chill. Okay. With proviruses, though, we're going to watch what happens. So with a provirus like HIV, we're going to ignore the fact that it's going to go from RNA to DNA. We're just going to assume that's already happened. And I'm going to end up with my nice chunk. I'm going to draw it really thick here. So this is a chunk of HIV DNA. Okay? And it's now a provirus. So I'm just going to say HIV DNA. So that's the HIV genome. Sorry, it's a little hard to see. And then we're going to take this and we're going to integrate it. Okay? So that's the term that I said before with bacteriophages. When they take their genome and insert it into a bacteria's genome, they have integrated it. And that's what proviral latency is. Not all latent viruses do that. Some of them are going to follow the episomal style, where they're going to just kind of keep their genome hanging out nearby. It's still going to get replicated, but it's just kind of hanging out nearby, not really messing with anything. But I like to think of the host genome that's just kind of sitting there and looks over, and there's like this weird little creepy thing that's sitting there looking at him. It's like, hey, are you supposed to be here? Well, I'm busy, and then you just keep doing your thing. But proviral latency is different. So it's going to take its chunk of DNA and it's going to integrate it. And so we're going to come over here, and now I'm going to just draw like that. We would open up that host DNA and insert the viral DNA into the host, and it becomes integrated. There's an entire enzyme devoted in HIV to actually doing that, integrase. Okay? So HIV found it prudent enough to evolve an enzyme whose entire purpose, they always have secondary purposes, but its main purpose is to actually integrate that. And that's one of the enzymes that we can actually inhibit. We can use integrase inhibitors to actually treat patients. Okay? So that's the difference. That's what we're talking about here. So when you see episomal latency versus proviral latency, that's the difference. Episomal is its own separate genome that does not get integrated, whereas proviruses have a genome that gets integrated. Okay? So let's zoom back in to this. And so this comes from that same review article, really good. So you can see here we've got our episomal genomes. So here are a bunch of examples of viruses. Epstein-Barr virus. So lots of humans have this. Most of these are herpes viruses, you can see. Essentially all herpes viruses. I mean, it says most herpes viruses here, but essentially all herpes viruses are going to undergo episomal latency. So they have that initial infection. Think of like if you got a herpes infection. So you get that cold sore on the surface epithelials, and then it's going to actually infect the nerve cells that innervate that area, and then it's going to infect that nerve cell. And I have a picture of that later on. Most of these, so that's how you're going to see your herpes viruses. I'm oversimplifying. That's your herpes simplex viruses, whereas you head for some other ones. So Epstein-Barr virus, Kaposi's sarcoma herpes virus, cytomegalovirus. Most of these are actually in the herpes virus family. Chickenpox, varicella zoster. Yeah, those are going to use their episomal infections. Now, there are some other ones. Some of these are a little weird. There's some strange ones. Let me get the name right. When we actually see proviral integration, we see those occur in a couple different ways. They're obviously different viruses. So here they're showing a couple of these. HHV6, which is human herpes virus 6, there's 6A and 6B. Those are viruses that engage in telomeric integration. So it's still a provirus. It's not an episomal infection, even though it is a herpes virus. Okay? It's more of a proviral approach. It's still double-stranded DNA. And what it likes to do is it has little tags on the ends that look like the repeats for the telomeres at the end of your chromosome, and it actually inserts in there. If you read the review article that this comes from, it's actually poorly understood why they do that. It probably has something to do with getting replicated. There's clearly some advantage, otherwise they wouldn't do that. The other two I had to look up to see what these were, because I was like, MDV and GAV2, what are those? So if you're a veterinarian person watching this, then a veterinarian person, really? If you're super into chickens, then you might know what MDV is. That's Marek's disease virus. I have to look at my cheat sheet. That's Marek's disease virus. It's just a virus that infects chickens. So you might encounter that sometimes. And GAV2, with that lowercase a, that was very strange for me. I had not seen that. It's gill-associated virus 2. So it infects fish. And we do see, there are herpes viruses and herpes-like viruses that infect every creature on the planet, including fish. And those can actually cause a lot of damage. Yeah, really interesting stuff. Now, on to the human T lymphotropic viruses, of which HIV sort of used to be lumped together with that, but we're going to separate it out. where we see some random integration. Now, it's argued some viruses, HIV appears to, at least most of the time, have random integration. So its provirus will be randomly integrated. And so we don't see a ton of, there's not a strong association between HIV and, let's say, cancer. Okay? There are other proviruses that can have a stronger association. And the association that I'm getting to would be if I took my provirus and integrated it into like a tumor suppressor gene or an oncogene, and I mess with that gene, mostly the tumor suppressors is what I'm thinking of. So if you took that provirus and shoved it into the gene for P53, now my cell can't engage in preventing cancer as easily, and so you can more likely develop a cancerous cell, which would not be good, okay? But if you're having random integration, well then every so often, yes, that does happen. But statistically, there's no real association with that. So that's a good thing. However, we don't really want these to be integrated at all, right? And so we need to prevent that. Now, the other concept that gets talked about, really interesting, is if you take that provirus, okay, it's going to be sitting over my shoulder here. If you take that provirus and integrate it into the host genome, it's just like a lysogenic cycle. It's just hiding inside the genome. The host cell machinery has no idea that it's there, and it's just going to replicate along with the rest of the host cell every time it replicates. So you're going to end up with, you could have one cell infected, and then every daughter cell after that is going to have that viral provirus integrated into it. That's how we see some relics. There are some relics of retroviruses that exist within our DNA. If you actually look into the genomic sequences, you can see that there are these carryovers of proviruses that have just survived. They never reactivated, and then they got kind of broken, and they're just kind of sitting in our DNA. I would argue that they may serve a purpose. We just don't know it yet. Scientists love to just make blanket statements and say, oh, they're vestigial, they're pointless, they're just these carryover things. And I don't know. In my experience, they probably have a purpose, but hey, I'm just a scientist too. So, how do we keep an episomal virus replicating within the host? Because it's not integrated, right? That's something that's reserved for the provirus for the provirus latency. So episomal latency, how it actually maintains that is really, really interesting. Basically, what you're going to do is you're going to tether your episome to the chromosomes. And so that's what you're seeing in this picture that's right above my head. So you can see we've got human papillomavirus up there, Epstein-Barr, Kaposi's sarcoma, herpes virus. And so you can see that they're tethering to the actual histones, and it depends on which ones, okay? But it doesn't have to be histones either, but that's just the most obvious one here, like H2A and H2B. It's going to tether itself to those during metaphase. And so when you have prophase, metaphase, anaphase, telophase of your actual cell replication, that's when you're like, oh, I need this guy to carry on. And so I'll take my episome, I'm going to attach it onto that metaphase chromosome right before it separates during anaphase. And then when that separation occurs, it can kind of keep on going, and it just kind of tags along. Really, really interesting stuff. Very difficult to study. So there's a huge gap in the literature for actually how this kind of stuff works, which is pretty cool. I always like the gaps in the literature. We're going to see so many gaps in the literature with viruses in general, which is one of my favorite things. I think it's pretty cool. Sorry, I'm looking at my cheat sheet here. So once the virus is in there, then it hangs out. Okay. So when does it reactivate? Well, that's a great question. I'm so glad you asked it. There are so many different reasons. One of the easiest ways that I like to talk about this is to think about if you've ever had a cold sore, okay, which is herpes virus. It's okay. People get them. 90% of all humans have herpes viruses like cold sores, so it's okay. I had Epstein-Barr virus, so that's integrated into my B cells. It's just hiding out there. Carried it with me. It's my buddy. So when is it going to reactivate? Okay, when are you going to get that cold sore? Well, think about if you've ever had a cold sore, when does it reactivate? Stress, right? Final exams. You're getting married. something stressful is happening in your life. That's often what will do it, right? My friend, chickenpox, varicella zoster, is the same way. So it'll reactivate and then it can cause shingles. And my friend, his wife, I was in their wedding, and she had shingles the month before their wedding. So on her back was a big line of because it follows back out that nerve and goes back out. So she had chickenpox as a kid. She didn't get the vaccine. And so the chickenpox virus reactivated and then infected the epithelial cells at the end of that nerve where it was innervating. Really interesting. And so it's poorly understood, you're going to hear that a lot, it's poorly understood why these viruses will reactivate. But we do know a lot about how a lot of these viruses will actually work. And that's something I want to talk about because sometimes we may want to actually reactivate them. With HIV, that's been a big push for how to actually treat people who have HIV. So let's talk about that. This is kind of a synopsis in this review article of the different things that can cause reactivation. So just think about some of these. So co-infections, that could reactivate it. I like to think of all of these as kind of stressors on the cell itself, on your body, which is stress on your cells. And so you're going to see, you could have a viral infection of a different kind or a bacterial infection that could stress you enough to actually initiate it. You could have just pure metabolic stress. You could have a big influx of inflammatory cytokines, which could be caused by stress. I'm just going to say stress all the time. You could add butyric acid. We actually used to add, when I was in my one of my labs that I was in grad school for, I was there for 4.5 years. It was an HIV lab. And so we dealt with latent viruses all the time. And we were actually using viruses as not HIV, but we were using an adenoviral vector as a gene therapy delivery vehicle. And we would add butyric acid. It smelled like butter. It was really gross. We had it in the fume hood, and it smelled like rancid butter. That's why I always associated butyric acid with butter. I don't know. Like they kind of sound like butter. And so I remember just pulling it up with the pipette, and it smelled so gross. But we would add a little bit of butyric acid, and it would rapidly increase the amount of virus that we would generate. It was crazy. And it had to do with the histones, and I want to talk about histones and HIV in a second. But there's just lots of things you can see. You can dig a little deeper if you want to go read this review article. They're going to go a little bit deeper into the actual research behind these. But something to keep in mind, there are multiple research groups, researchers around the country and around the world that are studying one of these little things on here. And so that's kind of crazy. A lot of money gets put into this. This is basic research, but it's really important because if we don't understand some of these basic ideas, then we might not be able to develop a treatment later on. Pretty cool. So let's talk a little bit about HIV. It's story time. So HIV is a proviral virus that gets integrated, right? And so one of the tricks with HIV is how do we know which cells have the provirus in them? How would you know? It's hiding in there. It gets sent into the daughter cells. How do you actually establish a latent infection? Well, that's a good question. What we usually see is that HIV will infect either T helper cells more often, okay, because they have CD4 plus a co-receptor. They can also infect macrophages. Okay? So people don't talk about that. CD4 T helper cells, CD4 positive T helper cells are the cells that we typically associate with HIV, but they can infect macrophages because macrophages can express CD4 and CCR5. I'm not 100% sure about CXCR4, I'll have to look that up, but I know they can express at least both of those, and CCR5 tends to be the more common co-receptor. And so you can infect macrophages, and when those get infected, the macrophage can actually then traffic into your brain because we do have macrophages all over your body and in all of your tissues. And so they can head into your brain and be those microglial cells, and they can hide in your brain. So not only is HIV hiding in other parts of your body, the reservoir can be in other parts of your body, but it can even be hiding behind the blood-brain barrier in your brain. And so you can get reactivation of virus in the brain. You could eliminate the provirus from everywhere else in the body, but if you don't get those little brain macrophages, you could still get HIV to reactivate. There are some really interesting things. I'm just going to talk about HIV a bunch. There are some really interesting ideas behind HIV and craziness, if that's an appropriate term, neurological symptoms of HIV. It's not super well understood. Now is not the time to go into it, but it's an interesting subfield of HIV. Whether or not that's associated with those macrophages, good question. Now, normally HIV likes to infect T helper cells. We're ignoring those macrophages. So those T helper cells get infected. And here's what I want you to imagine. Imagine you have the flu, and you have, maybe you're recovering from the flu, and you have a bunch of T helper cells, and their job is to fight off the flu. And they're doing a great job, but there's a ton of them around. And those T helper cells are hanging out, and you have some kind of exposure to HIV, and HIV loves T helper cells, and it infects some of those flu T helper cells. Okay, so now these T helper cells who are designed to attack the flu have now been attacked, infected, and their provirus for that HIV is now integrated into their genome. Well, what happens at the end of an infection, so let's say you've cleared all that influenza virus, or let's make it coronavirus now. Now we'll stick to influenza. So you've cleared all that influenza virus, so now your immune system starts to drop down a little bit. You have your PD-L1 upregulation, your CTLA4 upregulation to kind of suppress that immune response back to a more normal level. Okay, and so as that immune response drops to a more normal level, though some of those T helper cells will just die off. Same with B cells, the ones that have been doing a lot of the work, those plasma cells, those superactivated T helper cells, superactivated is a poor term, they either die, but some of them will remain memory cells. We have B cell memory cells, we have T helper cell memories, we have T killer cell memory cells, and some of them will enter such a powerful memory state, we call them resting. They don't do much. They're alive, we know that much, and they're not really doing a whole lot. There's not a lot of, they're not replicating, we know that as well. And so when you have a resting T helper cell, they're not doing anything. They're just hanging out. So imagine if some of those T helper cells that were fighting off the flu and they fought it off, they became resting memory cells, but they got infected with HIV. So that provirus is now hiding inside the chromatin of that resting T helper cell. That resting T helper cell can traffic all over the body. It's hanging out in lymph nodes and your liver and your spleen. It could go anywhere. Who knows? When does it reactivate? Great question. When you encounter the flu again, right? Because that's its job. It's a T helper cell that's a memory cell that's remembering influenza. So when it sees influenza again, boom, it reactivates, jumps back up, and the HIV that was hiding inside there, he's super excited. I don't know why it's a he. It gets super excited and it's like, oh my gosh, this cell just reactivated. Awesome. I'm going to start doing my thing. And it starts taking over the cell, making HIV. And now you have a massive uptick in HIV. So that's just one sneaky way that HIV likes to hide inside of your cells, inside of resting T helper cells. So eliminating that resting T helper cell reservoir is really, really important. And I actually worked in the Silicano lab for a while. So he's one of the most famous people for working on the resting T helper cell latency problem, that reservoir problem. Basically saying that the half-life of that reservoir is so long, like 75 years or something, that it's essentially too long to ever cure someone of it. Unless you could find a way to actually activate those cells and get them to exit that resting state, and then you can see that there's virus in there. So if we have a patient on antiviral drugs, awesome. So those drugs work against actively replicating virus. That's how they work. So if your virus isn't doing anything, it's just a chunk of DNA. There's nothing to distinguish it. So what we need to do is get that chunk of DNA doing its thing so that we can actually see something. It's almost, the DNA is like the genotype, and we need something phenotypic to actually see it. And so what we'll do, and this was a concept that people were throwing around for a while, and they mentioned it a little bit here. It shows HDACI right there. So that's HDAC inhibitors. So we need to understand a little bit about chromatin structure and how cells actually replicate their chromatin. And so you may know that histone, you have your DNA, and then you have your histones, and wrapped around your histones is your DNA. It's a nice way to keep it nice and organized, okay? That's what you're seeing in blue on this picture, where it says butyric acid and HDAC-I. That's your histones, are the little circles, and then the blue, darker blue is wrapped around them. So when you acetylate your histones, okay, it causes them to expand. Okay. So when I take an acetyl group and I stick it on there, I don't know why it's acetylate, not acetylate. I guess you could say it either way. When you acetylate, it causes them to expand. I'm oversimplifying things a little bit here, but that's basically what it means. So when you acetylate your histones, they expand out, and it lets me do transcription easier. Okay? So if I deacetylate, they close back up, so it's more condensed chromatin. Okay, so acetylate, deacetylate. So if I inhibit that process, so histone deacetylase, okay, so histone deacetylase will, if I acetylate, it goes this, so a histone deacetylase enzyme will cause the chromatin to condense, right? Which is totally fine. Sometimes you want your chromatin condensed down so that you're not really using it for transcription. Sometimes you want to open it back up, okay? That's fine. That happens all the time. This expansion and contraction is happening all the time in the cell. Now, if you're HIV, you probably want to hide most of the time. If you're not actively replicating, then maybe you want to hide out and you want to be a provirus for a while. And so you would love to have those histone deacetylases close up the chromatin that you're inside of. Okay? We see that the TAT protein, transactivator protein in HIV, influences HDACs, okay? Which is very, very strange because it may be able to do that while it's actually a provirus. It's very strange. If you want to dig way deeper into the literature, go for it. We're not going to talk about that today, but suffice to say, HIV itself has a protein that will interact with histone deacetylases to cause chromatin to condense down because it's going to hide the provirus, right? We don't want it nice and open. If I'm an HIV provirus, I might want to hide for a while, just kind of hang out, just let things kind of chill out a little bit. The cells have a lot of weapons to attack retroviruses, and so I don't want to be replicating right now. And so if I get nice and condensed down, I influence those histone deacetylases to be nice and condensed, awesome. So it was proposed, and we've been testing it for a while now, to use histone deacetylase inhibitors. So if histone deacetylases condense down the chromatin, if I inhibit that, then hypothetically it should open it back up. And so that's the idea behind a number of drugs. There are at least 4 that I can think of that have gone through clinical trials. And what you'll do is you'll take a person and put them on anti-HIV drugs. They have HIV, but that reservoir is still hiding there. It's really hard to get rid of. And so what we said was, all right, let's give them this drug, this HDAC inhibitor. So histone deacetylase condenses it down. So if I inhibit that, if I add a histone deacetylase inhibitor, it should open it back up, allow those proviruses to activate, get transcribed, thus making HIV proteins, which I can then attack with the drugs that the patients are already on. So that's been the proposal to actually eliminate HIV latency. Of course, it's not that simple. You have to make sure that those drugs get everywhere. You have to make sure that you are reactivating every single infected cell with a provirus, which that can get complicated. Like I mentioned, what if it's one of those macrophages hiding in your brain? So now this is where the pharmacokinetics and pharmacodynamics of the drugs that you're using, you have to make sure that they're getting to the right places for long enough. And so it gets really complicated really fast. But hey, that's why pharmaceutical companies get paid a bunch of money, I guess. Let's make it political. But there are lots of clinical trials. They've had some success, but you're not hearing about it a ton because, I don't know, it's just hard to do. So that is viral latency in a nutshell, though. There are some other examples. One picture that I did want to show was the idea, and I mentioned it earlier, of let's say you got a herpes virus infection on your face, and it's going to infect the nerve cells that innervate that area, and it passes down and infects the nerve itself. Now, something really interesting that happens is usually when herpes viruses reactivate, so you get that infection, that initial infection, it hides out, you don't have cold sores anymore, but you get under stress, those cold sores reactivate, they come back out, and they are infectious. You're making virus there, okay? Sometimes the virus can go the other way. That's really scary. So chickenpox has been shown to do this sometimes, where usually it goes back out and causes shingles. But sometimes it goes the other way, and it can actually kind of sneak and infect some of the nerve cells where they're touching, because the nerves, it's one big long chain of nerve cells, right? So you have this initial infected nerve cell, it can infect the next nerve and then go into your brain. That's not good. That'll kill you. But hey, not to scare you, because it doesn't happen that often, okay? But it's just one other thing that's tied to latency. So hopefully that helps explain latency. The important ideas that I want to hammer home: the idea of an episomal latency versus a proviral latency, the difference between those, and the idea that a provirus can just hide inside of a cell for essentially forever until that cell basically gets reactivated.